Good morning students. Today we go through the lesson Principles and Process of Biotechnology in Plus 2 Biobotany. 5 more questions of this lesson are: first question is, define bioreactor, explain the two fermentation process in detail. Answer for this question is given in page number 69 and 70. In page number 69, define bioreactor. Under the heading Methods of Biotechnology, it is given in the second paragraph. Bioreactor is a vessel or a container that is designed in such a way that it can provide optimum environment in which microorganisms or their enzymes interact with a substrate to produce required product. The next is the two processes involved in fermentation are below that paragraph: upstream process and downstream process. Upstream process means all the process before starting of the fermenter, such as sterilization of the fermenter, preparation and sterilization of culture medium, and growth of suitable inoculum are called upstream process. Downstream process means all the process after fermentation is called downstream process, and the process include distillation, centrifugation, filtration and extraction. Now close the bracket. The second question of this lesson is, explain the fermentation procedure with the diagram. Students, for this, if they ask define fermentation means, in the same page number 69, first paragraph, third line, fermentation refers to the metabolic process in which organic molecules are converted into acid, gases or alcohol in the absence of oxygen. That is enough. Close the bracket. Then procedure with a diagram. Fermentation procedure, that is given in page number 70. Under the heading procedure of fermentation, there is given A to F, 8 points. You should write that 8 points, and above that one diagram is there, no, bioreactor diagram, that also you should draw. Then next question is, fourth question, given account of single cell protein. Answer is given in page number 70-71. Students, under the heading single cell protein, 4.2.2. First of all, you should write define single cell protein. That also you studied for your 2 mark and 3 mark questions. SCP or single cell protein define. That 2 mark you should write. Then next is how to write bacteria organism used for the production of single cell proteins are, it is given in page number 71. Bacteria: Methylophilus, Methylotropus, Cellulomonas, alkali genes, fungi, Agaricus campestris, Saccharomyces cerevisiae, Candida utilis, and the algae are Spirulina, Chlorella, Chlamydomonas. Then next is, close the bracket. The next is why world people won't use the single cell protein, or these proteins are having high concentration of amino acid and proteins. Single cell proteins are rich in amino acid and protein, so it won't digest easily. It will cause some allergic problems. That also you should write. It is given in the third paragraph. Then next is materials required for the growth of single cell protein are waste water from potato processing plant, straw, molasses, animal manure and sewage. Then close the bracket. The next part of the question is application of single cell protein. Under this heading, it is given 6 points. It is used as protein supplement, from that to leather processing as foam stabilizers. Close the bracket. The next question, fifth question, list out the steps involved in recombinant DNA technology. Students, for this answer is given in page number Recombinant DNA Technology, page number 71-72. Page number 71. First is Recombinant DNA Technology. For that you should write what is recombinant DNA technology. It is given under the topic 4.3.1 genetic engineering. Genetic engineering or recombinant DNA technology or gene cloning is a collective term that includes different experimental protocols resulting in the modification and transfer of DNA from one organism to another. The next is steps involved in recombinant DNA technology, or it is given at the bottom of the paragraph, page. First one is isolation of DNA. Above that 3 lines, you should write the heading insert. What is insert? Isolation of a DNA fragment containing a gene of interest that need to be cloned. This is called insert. Then below the second paragraph, you should write rDNA. rDNA is produced by the insertion of DNA fragment into a carrier molecule called vector that can self-replicate within the host. Then above the third paragraph, you should write, that is given in the page number 72. Write the heading host cell. Selection of transformed host cell is carrying the rDNA and allowing them to multiplying the rDNA molecule. Then in the last paragraph, you should write protein produced. The entire process that generates either a large amount of rDNA or a large amount of protein expressed by the insert. Then last paragraph you should write at the top side heading PCR. Wherever vectors are not involved, the desired gene is multiplied using PCR technique. The multiple copies are injected into the host cell protoplast, or it is shot in the host cell by host cell protoplast by shotgun method. Close the bracket. That's all about rDNA technology. Steps involved in rDNA, recombinant DNA technology. The next sixth question is draw the diagram of steps involved in rDNA technology. Answer is given in page number 72. Page number 72, the top of the page, steps involved in rDNA technology diagram, figure 4.3. You must draw that diagram for this question. That is very, very important diagram. And the steps involved in recombinant technology also important question. SCP important question, single cell protein. The next question is list out the properties of vector. List out the properties of vector. It is given in page number 74. Given in page number 74, properties of vector. Students, write down the page number. It is given in page number 74. Properties of vector. Under the topic 4.4.4 vector, second paragraph. Vector, first one is what is vector? Vector are able to replicate autonomously to produce multiple copies of them along with their insert DNA into the host. First is what is vector. Then next, it should be smaller in size. From that, you should open the bracket till vectors should have unique target site for integration with DNA insert and should have the ability to integrate with the DNA insert it carries into the genome of the host cell. Close the bracket. That is, the points of these questions are 4 points. In the fourth point, you should write 4 lines. First 4 lines. The next question is define vector, explain the different types of vector. Page number 75. Answer is in page number 75. For this question also, you should write define vector. For that, under the heading, page number 74, under the heading 4.4.4 vector, from the second line, in that paragraph, second line. A vector is a small DNA molecule capable of self-replication and is used as a carrier or transporter of DNA fragment which is inserted into the cloning experiment. Inserted into it for cloning experiment. Close the bracket. Then the types of vector. Types of vector are, first one is plasmid. It is given in page number 75. Under the heading types of vector, plasmid is given. Plasmid are, first you should write definition for plasmid. Plasmid are extra chromosomal self-replicating double-stranded, DS means double-stranded, circular DNA molecule found in the bacterial cell in addition to the bacterial chromosome. close the bracket. Then next is PBr 322. PBr 322, from the third line you should write P denote plasmid, BR respectively Bolivar and Rodriguez, name of the scientist who discovered the plasmid, and the number 322 is the number of plasmid developed from their laboratory. The next question is TI plasmid. Sorry, not next question, the same question. Next type of vector is TI plasmid. For that, TI plasmid is found in Agrobacterium tumefaciens, close the bracket. Then from the third line, the plasmid carrier transfer gene which help to transfer tDNA from one bacterium to other bacterium or plant cell, close the bracket. And next question is, that's all for vector. Define vector and explain different types of vector. Then ninth question, explain the various methods used in vectorless gene transfer. For this, the answer is given in vectorless gene transfer. For this, the answer is given in next question is define vector, explain the different types of vector over. Then explain the various methods used in vectorless gene transfer. Various methods used in vectorless gene transfer. For this question, the answer is in page number 76. Take page number 76. Direct or vectorless gene transfer method, it is given under the heading 4.5.1 direct or vectorless gene transfer. In this, vectorless gene transfer means the foreign gene of interest is delivered into the host plant without the help of a vector is called vectorless gene transfer. Close the bracket. Then the different methods are, first one is cell mediated gene transfer. From that you start the answer for this question. And second process is micro injection. For that also you should write a definition. Electrophoresis method of gene transfer. This also you write the definition. Liposome mediated gene transfer. This also you should write a definition. And biolistic method. All this topic you all this heading you already studied for 2 mark and 3 mark question in the previous video. Next is 10th question. Explain Agrobacterium mediated gene transfer in plants. Page number 77. Agrobacterium mediated gene transfer in plants. In page number, students, in page number 77, under the heading 4.5.2 indirect or vector mediated gene transfer, in that gene transfer is mediated with the help of a plasmid vector known as indirect or vector mediated gene transfer. For this example is TI plasmid from Agrobacterium tumefaciens, 4th line, is extensively used. This bacterium has a large size plasmid known as TI plasmid, and a portion of it is referred as tDNA. This bacterium has the natural ability to transfer tDNA region of its plasmid into plant genome. Upon infection of the cell at the wound site, it is known as natural genetic engineering. It is also known as the natural genetic engineer of the plant. The next paragraph also you should include. And the diagram at the bottom of the page 77, there is given Agrobacterium mediated gene transfer in plants on figure is given 4.14. That also you should draw for this. The next question is 11th question. What are restriction enzyme? Give different types of restriction enzyme and its role in biotechnology. Page number 72. What are restriction enzyme? page number 72, under the heading 4.4.1 Restriction Enzyme. At the top of the paragraph, you should open the bracket. The two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli was isolated in 1963. One was the enzyme which added methyl group to the DNA, while the other cut the DNA. The enzyme which cut the DNA is called restriction enzyme, endonuclease. It is classified into two types. The second paragraph, last line, that are exonuclease and endonuclease. Exonuclease are enzymes which remove nucleotide at one at a time from the end of a DNA molecule. Endonuclease are enzymes which break the internal phosphodiester bond within a DNA molecule. Then there are three types of endonuclease, restriction endonuclease. They are type 1, type 2, type 3. It is given in page number 73. Second paragraph, type 1, type 2, type 3. Only type 2 enzyme is preferred for use in recombinant DNA technology. Then next paragraph, the restriction enzyme hint to always cut a DNA molecule at a point of recognition and a specific sequence of 6 base pair. The sequence is known as recognition sequence. That is enough, students. That is enough. Close the bracket. Then next is 12th question. Given account of application of biotechnology. It is given in page number 88. Students, take page number 88. Page number 88, under the heading 4.8, application of biotechnology. There it is given 11 points. All the 11 points you should study, and please write 10 points for the question. Application of biotechnology. The next is, this is also very, very important question. The next question is, list out the different types of bioremediation technologies. What is bioremediation? It's given in page number 86. Page number 86. What is bioremediation? Definition. Answer is given in page number 85. Please turn page number 85. Under the heading 4.7.11 bioremediation. Come to 5th line. Bioremediation process is, it is defined as, first line, it is defined as the use of microorganism or plants to manage environmental pollution. Close the bracket. The next come to the 4th line, 5th line. Bioremediation process applied to the removal of oil, petrochemical residues, pesticide or heavy metals from soil or groundwater. Close the bracket. Then come to the next page, page number 86. Different types of bioremediation are phytoremediation, mycoremediation, bioventing, bioleaching, bioaugmentation, composting, rhizofiltration, rhizostimulation. Students, all these questions, all this topic you have studied for your 2 mark and 3 mark question. That also enough for this 5 mark. So there are how many types of remediation means? 8 types of remediation you should study, and one more point you have to study is what is bioremediation. You should write. The next is limit. What are the limit of bioremediation? They are given 4 points. That also important for the 3 mark question already studied. Next question. Tabulate the difference between blotting technique. Page number 81. Students, please take page number 81. Blotting technique. In page number 81, on tabular column is given difference between blotting technique. Southern blotting, northern blotting, western blotting. That tabular column you study is also very, very important question. The next is, list out the steps involved in southern blotting technique. Page number 80. For this question, page number 80, Figure is given now, 4.19 steps involved in southern blotting technique. For that, these steps you have to write all the steps from 1 to 8 steps. It's given 1 to 8 step, all the steps you should write. The next is 16th question. What is electrophoresis, and explain the isolation of genetic material using gel electrophoresis. For this, answer is given in 78 and 79, page number 78 and 79. Electrophoresis. First is definition electrophoresis. It is given in page number 78. Under the topic 4.6.4 molecular techniques. Isolation of genetic material and gel electrophoresis. It is given. Define electrophoresis. Electrophoresis is a separating technique used to separate different biomolecules with positive and negative charges. Close the bracket. Then explain the isolation of genetic material using gel electrophoresis. It's given agarose gel electrophoresis in page number 79. One paragraph is given. That paragraph you should write. Mainly used for purification of specific DNA fragment. Point number 1. Agarose is convenient for separating DNA. Third point, polyacrylamide is preferred for the purification of smaller DNA fragments. The gel is complex network of polymeric molecule. DNA molecule is negatively charged molecule. Under an electric field, DNA molecule migrates through the gel. Fourth point, the electrophoresis is frequently performed with marker DNA fragments of known size. Fifth point, the advantages of agarose gel electrophoresis are that the DNA bands can be readily detected at the high sensitivity. The bands of DNA in the gel are stained with ethidium bromide, and DNA can be detected as visible fluorescent illumination in UV light will give orange fluorescence, which can be photographed. Close the bracket. The next is name the methods used for screening recombinant plasmid. Explain that method. Answer is given in page number 78. Page number 78. What is screening? What is screening is given in page number 77 under the heading 4.6 screening of recombinant. Open the bracket. After the introduction of rDNA into suitable host cell, it is essential to identify those cells which have received the rDNA molecule. This is called screening. This process is called screening. And the different methods used in screening are: first point, blue-white colony selection method. Second point, antibiotic resistant marker. Third method is replica plating technique. First, blue-white colony selection method. For that, powerful method for used for screening is this method. In this method, reporter gene LAC-Z is used. LAC-Z encodes beta-galactosidase and contains several recognition sites. Beta-galactosidase break a synthetic substrate called XGAL. If a foreign gene is inserted into LAC-Z, the gene will be inactivated. Therefore, no blue color will develop. Because beta-galactosidase is not synthesized due to inactivation of LAC-Z. White colored colonies on the medium contain XGAL, whereas the other cell contain non-recombinant DNA will develop the blue colored colonies. Next is antibiotic resistant marker. Next method, antibiotic resistant marker. Antibiotic resistant marker is a gene that produces a protein that provides cell with resistance to antibiotic. Bacteria with the transformed DNA can be identified growing on the medium containing an antibiotic. Such antibiotic are ampicillin, chloroamphenicol, tetracycline, kanamycin, etc. Next method is replica plating technique. A technique in which the pattern of colonies grown on the culture plate is copied. A sterile filter plate is pressed against the culture plate and then lifted. Then the filter is pressed against the second sterile culture plate. This results in the new plate being infected with a cell in the same relative position as the colonies on the original plate. Usually the medium used in the second plate will differ from that used in the first one. It may include an antibiotic or exclude a growth factor. Close the bracket. That's all. Enough for screening method. Different types of screening method. Blue white colony method, antibiotic resistant marker, replica plating technique. The next question is 19th question. Explain the insect resistance of BT crop. Page number 83. It is given in page number 83. Insect resistance of BT crop. Under the heading 4.7.3, insect resistant BT crop, BT cotton. BT cotton is a genetically modified organism or genetically modified pest resistant plant cotton variety which produce an insecticide activity to bollworm. From that paragraph, you should write above the advantage of the BT crop. For that is 3 paragraphs. The next is twentieth question. Explain the herbicide tolerance of genetically modified crops. Page number 83. Under the heading 4.7.2, herbicide tolerant BASTA. Define BASTA you should write first. Non-selective herbicide containing chemical compound phosphinothrysin. Then BASTA herbicide tolerant gene PPT was isolated from Medicago sativa. The plant that contains tolerance of this BASTA is having a gene called PPT and the plant name is Medicago sativa. Second point you should write this one. The next is PPT gene was introduced into tobacco and transgenic tobacco produced was resistant to PPT. Similar enzyme was also found in Hygroscopicus, Streptomycus hygroscopicus with a bar gene encodes for PAT and was introduced into crop like potato and sugar beet and transgenic crops have been developed. Close the bracket. The next is how RNA interference happens in nematode. Page number 82. RNA interference. For this, in page number 82, answer is given in page number 82 under the topic 4.6.10 RNA interference. And that come to the 20th line. RNA interference is a biological process in which RNA molecule inhibit gene expression or translation. From that point, you open the bracket till at the end of the paragraph. This RNA is seen in plant feeding nematode. Close the bracket. At the end of the paragraph. The next is 22nd question. Define blotting, uses of blotting, and explain different types of blotting. This also you studied for 2 more questions. Define blotting. Page number 79. Answer is given in page number 79. Define blotting. Blotting means, answer is in page number 79. Blotting techniques are widely used in analytical tool for the specific identification of desired DNA or RNA fragments from large number of molecules. No need of that line. From the 4th line. Blotting refers to the process of immobilization of sample nucleic acid or solid support. The blotted nucleic acid are then used as target in the hybridization experiment for their specific detection. Uses of blotting, it is given above definition. Above that definition, blotting techniques are widely used from the beginning of the paragraph. Widely used analytical tool for the specific identification of desired DNA or RNA fragments from large number of molecules. Then explain different types of blotting. Types of blotting it is given: southern blotting, northern blotting, western blotting, and what is southern blotting and steps. The next is what is Northern blotting. What is western blotting? Definition of this blotting. All the blotting you should write for this answer. The next this question. Next question is define target gene effect. Explain the two types of targeting vector used for gene knockout. The answer is given in page number 80. Page number 80. Target gene. Target gene is target DNA, foreign DNA, passenger DNA, exogenous DNA, or gene of interest, or insert DNA. That is to be either cloned or specifically mutated. Is given under the heading 4.6.6 bioassay for target gene effect. In that first 4 lines definition target gene. The next is explain the two types of targeting vector used for gene knockout. The two types of targeting genes are insertion vector. First one is two types of vectors are insertion vector. Point number 1. Insertion vectors are entirely inserted into target locus as the vector are linearized within the homology region. Initially, those vectors are circular, but during insertion become linear. It leads to duplication of sequences adjacent to selectable marker. The next is replacement vector. Next type of vector is replacement vector. As the homology region, it is collinear with the target. This vector is linearized prior to transfection outside the homology region, and then consequently a crossing over occurs to replace the endogenous DNA with the incoming DNA. Close the bracket. So students, so far we have seen 23 5 marks in this lesson, principles and process of biotechnology. In the 4th lesson of biobotany, we have seen 23 5 marks. All the questions are very, very important. Out of these questions, somewhat more important questions are single cell protein, SCP, given account of single cell protein. Then second one is recombinant DNA technology. Third one is various methods used in vectorless gene transfer. And fourth question is Agrobacterium mediated gene transfer in plants. Fifth question is application of biotechnology. Sixth question is electrophoresis and the isolation of genetic material using gel electrophoresis. And the seventh question, Bt resistant crops. These 7 questions are very, very, very important in this lesson. Students, please study these questions thoroughly. Write 2 or 3 times, and get ready for the examination. Thank you.